which is, well, you might know as well as a drug which used in Parkinson, which is inhibitor of monoamine oxidase, is enzyme which utilize dopamine. Well, it's responsible for producing of the signal in astrocytes, and the verapamil, which is a blocker of the potential sensitive channels, also can block calcium signal in neurons, but not in astrocytes. And this is the publications which been out on the basis of this. And okay, well, we establish the signaling role of dopamine and possible role in the neurodegeneration, and we test the scheme on the basis of the Parkinson model, and we found that again, well, we can see that, sorry, that induction of the cells with dopamine induce different kind of signal. This again, it's still control cells, and we can see calcium in cytosol and calcium in mitochondria. Mitochondrial calcium is very important because, well, we have calcium overload in mitochondria. It might induce cell death, and actually is a major trigger for neurodegeneration. And this is a pink one, and if we induce calcium signal here, we can see that mitochondrial calcium start to redistribute and become very bright. That strongly suggests that, well, even physiological calcium signal which induced by dopamine might be toxic for the cells and might induce cell death. That strongly suggests that for pink one deficiency, the dopamine is toxic. And we in the order to understand this, well, we measure the different parameters like activation of the reactive oxygen species, which is damaging for cells, and this is the mitochondrial membrane potential. And when we start to support cells with mitochondrial substrates, the situation becomes much better. Well, this is a activation with dopamine. This is, well, just substrates. You can see. Then we add the different substrates here. We'll restore this damaging which induced by reactive oxygen species being seriously depressed. And the same with mitochondrial membrane potential, which suggests that we have a problem with energy in the cells. And again, well, we tested this on the patient's fibroblast again, because, well, all what I told you before was done on the basis of the mitochondrial, pink one deficiency, suggesting, well, that the gene was just knocked out or knocked down. Here, this is a fibroblast of the patients, which finally been immortalized, and we tried to establish the basic principles, and it was control and patients, and we found using all this technique which I described before, we found that it's easy to find the pathology in the cells. Well, this is a red line, it's the mitochondrial membrane potential in response to calcium, and we can see that here we have no changes, and here it significantly dropped. And
the right hand side is the mutant.
Yes, and
Mutated.
this is a control. Sorry, it's a patient fibroblast with mutated pink one.
Mutated, yeah.
But again, well, we've been blinded for a long time, and then we had a scheme, and with table we found that what was very interesting, that it was a family of two, and in one case it was very early onset, 30 years old, and the sister had a latent set in her 70s. And what is important, they had the same mutation in PINK1, and the only difference between them, it was activation of one of the mitochondrial complexes. That suggests that, well, if we support cells with energy, we might protect neurons against neurodegeneration. And this is a very important observation which can be used in as a drug screen, and we can see here that we induce cell death in neurons with dopamine and try to protect them with number of the compounds which I mentioned, like verapamil and different mitochondrial substrates and antioxidant, and when found that most of them are very protective, especially mitochondrial substrates, again, which strongly suggests that we have energy deprivation in case of the PINK1 deficiency or PINK1 mutation. And when we support cells with energy, we protect them, and it's on the different level of the cells and the different cells. But we should remember that our neurons require much more energy than any other cells, including heart. And that's why if we have energy deprivation, the first cells which under danger of the cell death is neurons. And I'd like to point that this study push us to investigate one of the very strange and old compound which called polyphosphate. And we found, recently, because we still cannot explain on the basis of the modern science what is exactly and how it leads to neurodegeneration. And when we start to study this compound, we found first that it's a novel neurotransmitter, a compound which transmit signal between neurons, which is very important. But what is important from other point of view, that in PINK1 the level of this polyphosphate is significantly higher, and when we check all other Parkinson genes, it show the same phenotype. We had a much higher level of this polyphosphate, and it's actually the direction which we're walking now. And I'd like to point that, okay, this is probably my final slide of my fellowship, but thanks to the Parkinson UK, I'm working in the Parkinson, and thanks to the Parkinson UK, I've been triggered to this area, and I'm still working, using some other grants, but again, I'm not planning to be out of this research, and I'm in the time of submission of the new application to Parkinson with new projects, which I think is very exciting and which might potentially lead to some kind of therapeutic strategies. Sorry?
Thank you, and best of luck.
Yeah, thank you. And I'd like to, just quickly.
Sorry, tea copy for you?
Yeah, yes, please.
You want now?
Yes,
Yeah, please, yeah.
Yes, and I'd like to point that this grant is not just not just grant, and all the study is not just in our